and it's not known why the vast majority of plants are resistant to the vast majority of pathogens, and only it's the exception when a pathogen can cause disease on a plant, because each plant is being attacked by millions and millions of pathogens all the time, but only a few have evolved the ability to really cause disease on plants.
A lot of the work that we do in my lab focuses on food safety, looking at, in particular, these dangerous toxins that occur in food and feed, particular toxins that are produced by fungi. We're interested in crops such as wheat, barley, and corn, where these toxins, we call them mycotoxins, can accumulate. And as you know, these are staple foods that we eat, that domestic animals eat. And of course, the contamination of these staple foods with these toxins is a significant threat to human and domestic animal health.
I study parasitic plants, which are by and large a huge problem in other parts of the world. Not necessarily here in Virginia, but I have the freedom to be working on a world stage here in Blacksburg.
And that's one of the reasons why I joined this lab, to look at, because when you look at my country, for example, more than 50 to 60, sometimes even 80% of crop production are really devastated by this kind of disease caused by bacteria.
What we believe is going on is that pathogens are secreting dozens or maybe even hundreds of their own proteins to the interior of plant cells. Once they get inside, we believe that these proteins act as molecular saboteurs by targeting particular proteins that play key regulatory roles for plant processes such as immune responses, cell structure, or even metabolism.
And in particular, in trying to understand how did these bacteria evolve over time to become very dangerous pathogens of certain plants, and how do they cause disease? How do they overcome the very sophisticated immune system of the plants to cause disease?
We're interested in rapid detection methodologies to actually detect pathogens in the atmosphere really in a matter of minutes. We use technologies such as the unmanned aerial vehicles that you see behind you here to collect large volumes of air that might contain very dangerous pathogens. And we have techniques to actually culture these pathogens in the lab and try to understand what they're doing there, where they're coming from, where they're going, and ultimately being able to predict the movement of these pathogens in the atmosphere to healthy crop fields. In other words, a part of an early warning system for plant pathogens.
By understanding the mechanism, how these bacteria can infect, plant cell, I think I may be able to go back home and apply the same technique, I learned here, the same knowledge I have here to help, my country, to solve this kind of problem.
What you see behind me is a special quarantine facility that lets me work with parasitic plants that have come from Africa and the Middle East and cannot be allowed out into the state of Virginia.
Which, if you're growing subsistence crops, people aren't going to be able to use those fields for a period of time, and that means they're not going to be able to eat. And so if we can study this mechanism of parasitism, we can go and help these people to figure out a way to get rid of the parasite and still be able to grow their crops.
So we hope that by understanding what these pathogens that are able to cause disease on certain plants we study, what they've done differently that allows them to cause disease on plants as opposed to their relatives that can't cause disease on that plant.
And our hope is that by the end of this project, we'll understand exactly what plant processes are being targeted by these proteins. Once we have that knowledge in hand, then we may be able to design new strategies for engineering resistance against pathogens that cause destructive diseases in agriculture.